Hello everyone, welcome back to Dopamine Biology. Please make sure to leave a like and a comment. Do not forget to subscribe. Today we will be talking about RNA or ribonucleic acid and its significance in biological systems. Our genetic material is encoded in DNA. DNA is famous, but we also know about RNA or ribonucleic acid. So what is RNA and what is it good for? Quite a lot really. RNA is similar to DNA in lots of ways. It is a long chain of sugars linked together by phosphate groups. There is a cyclic base attached to each sugar, and the bases can pair with matching partners to make a double helix. I will talk more about this later in this video. RNA resembles DNA, but the helix is a bit twisted. And often RNAs are folded into complex structures that are stabilized by long single-stranded loops. Although RNA does not serve as the hereditary information in most cells, RNA does hold this function for many viruses that do not contain DNA. Therefore, RNA clearly does have the additional capacity to serve as genetic information. Although RNA is typically single-stranded within cells, there is significant diversity in RNA viruses. Therefore, RNA has great capability as a genetic molecule for some viruses. Measles, Rubella, and the Ebola viruses are single-stranded RNA viruses, and these viruses exploit all known mechanisms of genetic variation to guarantee their survival. Distinctive features of RNA virus replication include high mutation rates, high yields, and short replication times. RNA viruses have astonishingly high mutation rates, up to 1,000,000 times higher than their hosts. And these high rates are correlated with enhanced virulence. Rhinovirus, influenza viruses, including COVID-19, and the mumps virus carry RNA as their genetic material. Why do RNA viruses generally have very high mutation rates compared to DNA viruses? It is because viral RNA replication enzymes do not have the proofreading ability of DNA replication enzymes. RNA replication enzymes are more prone to make mistakes during RNA replication. RNA viruses mutation rates are almost disastrously high, and a small increase in mutation rate can cause RNA viruses to go locally extinct or be more infectious. The virus causing influenza is one of the best known pathogens found in various species. It's the best example of viral mutational changes. A human influenza virus could exchange genes with strains from different animals, and that is how pandemic strains arise. In particular, the virus is found in birds as well as mammals, including horses, pigs, and humans. The genetic diversity of RNA viruses is one reason why it is more difficult to make effective vaccines against them. Now, let's examine RNA's structure. RNA is typically single-stranded and is made up of nucleotides, just like all the other nucleic acids. The phosphate group is attached to the hydroxyl group of the fifth carbon of the ribose sugar. Each nucleotide in RNA contains a ribose sugar with carbons numbered 1 through 5. A base is attached to the first carbon. In general, nitrogen including bases of RNA are adenine, guanine, cytosine, and uracil. The RNA-specific uracil forms a complementary base pair with adenine and is used instead of thymine in DNA. The bases form hydrogen bonds between cytosine and guanine, between adenine and uracil. The phosphate group is attached to the hydroxyl group of the fifth carbon of one sugar and the hydroxyl group of the third carbon of the sugar of the next nucleotide, which forms a phosphodiester linkage. And an RNA polynucleotide may have thousands of such phosphodiester linkages. Even though RNA is single-stranded, most types of RNA molecules show extensive intramolecular base pairing between complementary sequences within the RNA strand. An important structural component of RNA that distinguishes it from DNA is the presence of the hydroxyl group at the second position of the ribose sugar. The subtle structural difference between the sugars gives DNA added stability, making DNA more suitable for the storage of genetic information, whereas the relative instability of RNA makes it more suitable for its short-term functions. It's partly the extra oxygen and partly the special ability RNA has to fold up into complex shapes to form tools that can do things, whereas the double helix is regular and stable. The DNA double helix holds information securely but does not do much else. Also, RNA has larger groups, which makes it easier to be attacked by enzymes. RNA's ribose sugar is more reactive because of the presence of hydroxyl group on the second carbon, as I mentioned before. DNA has smaller groups, which makes it harder for enzymes to attack DNA. Of the many types of RNA, the three most well-known and most commonly studied are messenger RNA, ribosomal RNA, transfer RNA, which are present in all organisms. These and other types of RNAs primarily carry out biochemical reactions similar to enzymes. In the next video, I will talk about the main types of RNA in more detail. Thanks for watching.